back to make sure I am in Calliopsis, which is a blue line. And let's look at the factors here. So most of these, with the exception of Protandrena, which hopefully we'll get to, are two submarginal celled things. So the interactions tend to be with each other in terms of ID problems because there's not as many groups that have just two submarginal cells. But we get into some different areas that we either haven't looked at for a while or are new. So if we go through the female, and we're going to look at a female in a second, we see two submarginal cells. This is, again, this basic set of characters, a trim set. It's not an absolute set. It's what I consider to be the fastest set to ID this a specimen to genus for that group. And so in some ways it's my favorites, and you certainly should modify it as you go along and create your own modifications to this. So if we're looking in here, two submarginal cells, two subantennal sutures, I believe that's a andrenidae-wide family characteristic. Okay, and so when looking at that, then that kicks us back to looking at all these small species and trying to not and getting rid of Andrena as a choice. So the inner suture shorter than the diameter of the antennal socket. So we'll look at that. So that's the inner suture of the two subantennal sutures. You can see the semicolons help separate out the facets there. And the other groups have longer ones. Facial fovea, so if you remember Andrena, the facial fovea there were relatively wide, and the inside of the facial fovea were covered with very short, let's call it fur-like hairs. And you would see that by rocking the specimen back and forth. The groups that we're looking at today also have facial fovea, but they're all narrow and lack hairs on the inside. And most of them are relatively short, but extremely narrow. The marginal cell, and this is something that helps separate this particular group out from Perdita, is relatively long, not long as in Xylocopa long, but long as in comparison to Perdita. We'll look and talk about that. And the abdomen has hair bands, which most of the others, oops, what did I do there? I think I just deleted it. Let's escape that. And so most of, there we go. Most of the other groups don't have hair bands on their abdomens. They have, they may have markings on their abdomens like Perdita often does, but they don't have actual bands of hair. So with that, let's jump to a specimen. And I should also point out, in general, all these groups that we're looking at have pollen-carrying hairs, but they're extremely sparse compared to some of the hairs that other groups have, like in Apidae, for example. You don't have these shaggy mop-like pollen-carrying hairs. So a lot of times you might look at a specimen in these groups and say, oh, I'm looking at a male because I don't see any pollen-carrying hairs. It's relative. So you may be looking at a female, but the hairs that carry pollen are so very sparse that they don't show up. Remember, these are small species, so sparseness probably is to their advantage to grab pollen that is going to be larger in comparison to their body size than some of these larger groups, larger bees like in the Eucerines and the Melisodes and things. So we will jump to a picture here. So we're looking at a female, and an interesting feature of, I think, all the Calliopsis is that the males and females have a very, very different look. So the females usually have very white markings on the face, and the males have, and relatively restricted, and the males have unbelievable amounts of bright fluorescent yellow on their bodies in several different places, and they often look like wasps, or is like, what am I looking at, kinds of things. So they for whatever reason, look quite different. And that's a general pattern. The males, I can't think of any exceptions. Someone can disabuse me of that one, where the males have less markings or fewer markings than the females. So in almost all cases, or all the cases I can think of, they have a greater amount of these pale markings. It may vary from white or cream to yellow to reddish to almost bright red. All right, so one vibe of Calliopsis andreniformis that is literally almost visible when you look at it with your eyes is these white markings form what amount to 3 stripes on the face. And so you can spot that, and you just, with a little bit of practice, you immediately know that you've got Calliopsis there. When you key it out, you have to go through a lot of questions within Discover Life to get to the ID because, as you saw from those shortcuts, there's no one thing that really defines Calliopsis from the others. So in this picture, I'm going to outline right now with the cursor the very short and relatively or very narrow facial fovea here. So there's the fovea. And, you know, you can't quite see, but there are no hairs to the inside. So all of these groups have something like that. This little slot, sometimes longer and narrower, but often just like that. The sizes and shapes usually are not that useful. In Hylaeus, which we covered already, I think we did, I'm pretty sure.
Yes.
It is a bit more useful, but in most of these others, despite differences in the shapes, it's not useful at a species level most of the time. But in terms of presence, it is. So let's shift now and look at these subantennal sutures that were mentioned. And let's see if I can rotate our specimen a bit more here and get us a good shot. So we're going to go to the base of the antennae, and we're going to look for, so the fossa, it would be called. So the fossa is, I believe it just means essentially hole. And we have a lot of brightness here. Let's see if I can take care of that. Not really. Doesn't seem to. I wish I knew how to decrease the shininess, let's call it, on this. But here's the antennal base. This one, the antenna is snapped off on this one conveniently. Here it is on, but same idea. There's the base. And then here's one antennal suture, and the antennal sutures are running from the antennae, antennal fossa, rather. So the hole in the head part to the clypeal rim. So here's the suture that defines the plate we call the clypeus. This is all clypeus here. And you can see this one's obviously longer than that one just based on architecture. And this length here is shorter than the width of the antenna fossa or base. And so that is an ID characteristic that we'll come back to in other groups within the Panergynes in particular, and that separates out Calliopsis. So I'm going to back this off a little bit. I believe we don't have any other head structures to discuss here.
Can you say something about what are the Panergynes?
So the Panergynes would be Perdita. Well, in the ones that I have specimens for would be Perdita, Panergynous, and Pseudopanergus. And there, elsewhere there's others, like there's a Panergus in addition to the Panergynous, just to be confusing. We're going to hopefully cover all those, at least if not today, another time. So they're all small. This is a group of small bees. And now let's flip to wing architecture here. And we'll talk about length of the marginal cell. So we'll zoom in. And there is our forewing. And in general, I'm not sure that's in the characters that were listed here, the tip of these Andrena, Panergyne, Protandrena, Calliopsis maybe, groups all have a tip of the marginal cell that's pulled away by quite a bit from the margin. If you're in the guide, and you're certainly if you're using Michener, or not Michener, but McGinley and Michener and Danforth's guide, this is a crucial couplet in the dichotomous keys that separates out some big groups, which is the apex, so the very tip, is separated from the wing margin by more than 3 widths of a vein. And it has several little subclauses, which makes it a real pain in the ass, frankly, character. And so we have it in there because it is useful, but it's confuses everybody. And when people take classes that use that. Additionally, the length of this submarginal cell, and I believe it just refers to the length along the margin, is greater than half the distance between the tip and the marginal cell itself. So if we take this, flip it over, that distance, which would be roughly there, is more than halfway. What that's doing is separating out Perdita, which is a pretty similar species. It's got a very short submarginal cell. So let's see, was there anything else that was mentioned in the shortcuts that we need to talk about? It's got relatively straight, so you can see a slight arch basal vein, so that also takes care of most of halictity. But the subantennal sutures, not my favorite thing, but when you get started, you have to look at them a number of times to get to the genus. After a while, you just look at this and go, oh, that's a Calliopsis. And don't forget that when you get more and more familiar, you can go to the Discover Life guide and just say, I want to only look at, andrenity. I only want to look at, the Panergynes, and it will recreate the guide for just that group. Well, again, I should go back for people who haven't been here in a while and just refresh how that works. And as you get even further down the line, you'll be like, oh, how do I tell like Panergynous from Pseudopanergus? And I know there's a couplet about that. So there's using the has button in Discover Life, you can go pull up every single question and then do a find to get to that particular couplet. So as you go along, you're not using the guide as a start to finish thing. You're using it to jump to sections that you know that you need to look at and refresh yourself. Is it a Eucera or is it a, Pepinapis, that kind of stuff. So I think we are done with the female here. Holler if I've left something off in terms of ID things, and we'll go to the male. It's over here. And you'll see how very different it looks. And this look is just really, again, one of these really unique things. It's just actually a little bit bizarre looking. I think everyone has this initial, like, what am I looking at here kind of thing. And we have a lot of excess light. Let's see if I can fix any of that with, boy, it just does not. Let me zoom way in and get the face filling up much of the middle, and then hit white balance, see if it will tone it down. Gosh, I wish I could control that. That may be as good as it gets. Okay, so let's, now that we're here, let's focus on the face. It has this very almost parrot-like look to the face. You can see this is really not showing as explicitly fluorescent yellow as it is. This is a little bit, it's all the same color. It's just reflecting yellows and oranges in photography, and in these kind of video feeds that are reflective are very what we call hot colors, and they often blow out in when we try to take pictures of them. All the things we suffer. So you can see everything below the antennae is yellow in this particular species, maybe in all of them, I'm not sure. We're going to look at, again, the subantennal sutures. Let's see if we can jump in there. Ooh, there's a little bit less noisy there. So we're at a slight angle, which is not bad. Here's the clypeus. This is between the two. This is the supraclypeus there. This is the clypeus. We are looking at the base of the antenna, so the fossa, where it goes into the head, and then we have the inner subantennal suture and the outer. So there's two. In lots of other groups, there's only one outside of andrenidae. And then this inner one is shorter than the width of the fossa. So that puts us in Calliopsis land amongst these other characters. I don't believe there is a fossa. If there is, it's a shadow of the female in the male. We'll just take a quick scan. Sometimes you see that the male, because it is haploid, gets to have little sort of configurations that are not useful, it seems, but are some shadow of what the female has, which is a useful thing. It has the other general characters of the two submarginal cells, the length of the marginal cell, which we'll see under various circumstances, is going to be very similar. I'm gonna run my finger under there. Wow, that blows it out. But you can see, I think, the wing veins pretty well. Let's change that. Oh, nice. So again, this length is more than half the length between the tip and to the end of the submarginal cell. So I think we are done also with that male. And we are going to jump over now and let's do Perdita. Since we have a couple characteristics that are Perdita alone thing, or it's a group that now we're into. Perdita is a group that a lot of people don't run into until they do. And so it's a relatively uncommon group. So, but again, you can be in an area where you can literally catch hundreds of these under the right circumstances when they all come out often in a sand, prairie, edge of river kinds of circumstances. I want to jump right now back to the memorization key. What am I doing here? There we go, and go up to Perdita, go in the female, jump over here. Are you following along? And oh, it went down to the males, and we click, it should open up everything. Okay, so again, two submarginal cells, two subantennal sutures. Inner suture in this case is longer than the diameter of the antennal socket, so we'll take a look at that. Facial fovea, relatively narrow, same thing basically as Calliopsis and the others. Length of marginal cell, so this is the key character here, in addition to actually the, in terms of the other panergynes, is the short marginal cell, the really stumpy, truncated one. And yeah, so we have a couple other characters, like sometimes there's a little triangular extra cell on some of the cross veins between the two marginal cells, submarginal cells, and some of these specimens, including this one, are slightly metallic, sort of very much in vein of Ceratina, and even more so with the Dialictus group of lazy glossums. And they're about the same size, small, short, size of a grain of rice. So we're going to look at Perdita octomaculata, the 8-spotted Perdita. You can see the spots here. Perdita in general has more colorations than the other panergynes. Is that true? Maybe not pseudobernergus, but certainly more in the female than others, unless they don't. So some of them are almost entirely dark. Key characters are the amount and distribution of the light areas in the abdomen.
Well, and then you have the super pale ones from the west.
And yes, in several parts of the country actually, but more in the west. But you can, it's they seem to be associated again with sand. That you can get a specimen. Oh, sometimes within the species, I believe, and other times they are separate species that are almost completely white. Would make sense if you're hanging out in sand areas. Okay, so this is the stigma. Look how big the stigma is in general. You know, we saw in the Eucerines, this was almost minute. This is very large, as it is in Andrenidae. And in comparison to the marginal cell, it's almost as large, perhaps is as large. 2 submarginal cells as per other groups. And you can see it's very truncated, so very chopped off. The tip is well separated from the marginal cell, and this distance here along the marginal cell on the edge of the margin of the wing is less, or maybe just roughly equal. So if we were to flip that over, which is how I do visually, it's going to end up somewhere right around there, and that's less than half the distance to the tip of the wing. So basically we're saying it's short, it's really short. It's shorter than the other panergynes and andrena, which are not super long, but they are way longer than this. This is your main character right there.
Could you move the cursor out of the way so I can take a screenshot?
Yep. Should have cleaned it up a little bit. So now, are you ready, Claire? Mm-hmm. Okay, so if we flip the specimen over, we'll take a look at some of the head characters for this one. And females often can have, but not all species have, markings on the face, but most of them do. I think we can play around with lighting a little bit here, but what we're looking at is here's the margin of the clypeus right there. Here's the fossa, or the hole that the antenna goes into. Here's one subantennal suture. Here's another subantennal suture joining with the suture that defines the clypeus. Again, this middle area is the supraclypeus, which we're ignoring. But this inner suture, let's see if I can go a little bit more in to there. The inner suture, the length of that, so the distance between the clypeus and where it joins, the junction, is as long or longer than the distance of the diameter of the fossa. So this is going to separate it from, in addition to other characters, from Calliopsis, and it puts it in the pocket of the other panergynes. So you can see, let me make the focus a little bit better, the facial fovea here. And if someone were to spend a bunch of time, I'm sure that these facial foveas here, again, relatively narrow, relatively short, vary enough that there would be useful in ID, but I've never seen anyone do that, maybe largely because there's enough confusion at the species level about the species, and people generally just have a, very small number, not a long run of any species. They seem to show up in onesie, twosie kinds of things. Onesie, twosie. Okay, so you can see maybe a little bit here that the background is a little metallic, so there are straight black groups of Perdita, and there are others like this Octomaculata, which is probably the most commonly run into one. It's a fan of sand, but it doesn't have to be that sandy. And it shows up in interior, not just on the edges or in dunes. And you can also see it does have pollen in its scopa, but you can also, I think, perceive that the pollen-carrying hairs here are not that dense. We zoom in even more. We'll see that the femur, I believe in all this group, but perhaps just this one, the femur lacks any pollen-carrying hairs. I guess that's not true in Andrena, but in this one, it's tibia. The action is on the tibia, and it's not much. So I think that actually covers the Perdita situation, and we'll go and take a look at a male. As with everything, the males have more more markings than the female of a particular species, but because the overall level of markings in any among species is so variable, it's hard to generalize what a male would look like. Oh, I think I've got another female here. Let me grab.
While Sam is searching, our chat has been really quiet today. Are we sure we don't have anything coming up? What is the phenology of Perdita, Sam?
So on average, they are fall species, but there's a whole slug of little exceptions. So there's a cool Perdita that is a willow specialist, like sandy, sandbar kind of willow things, Lake Ontario even, and it's out in the spring when willow are blooming. There's one that's more midsummer, a couple that are midsummer that are on special. They tend to be, again, specialists. So there's one that's a specialist on what is it? It's Physalis, so that can be out at several times of the year. But in general, fall. So here's the male, same species. You can see some metallicness. You can see that in general, it's got more yellow. The subantennal sutures are still there. I don't want to belabor this because the characters that you're going to use are pretty much the same between the sexes. I just wanted to illustrate the male in relationship to the female. So yeah, you can't even see the inner, there's some goop on this area, so you can't see this other subantennal suture, but it is longer than the diameter. Okay, so let's go to Protandrena.
Sam, you're just extra clear today.
Oh, nice. I've just had like 17 sessions of practice. All right, Protandrena, bigger on average than the others, but it isn't saying much. It's still not approaching honeybee size. We will go to, let's go to the identification section here. It's not a panergyne, it's its own group, its own subgenus. And we go to, I only have females, so I don't, I tend to give away a lot of the specimens. So for whatever reason, no males, but it should, again, should be very similar between the sexes. So in this case, we have 3 submarginal cells. Okay, so that clearly differentiates it from the other groups that we talked about, Calliopsis and Perdita and the other panergynes. But that puts us in the lap of other smallish things, like, for example, almost all the halictids. So a differentiation piece is that the basal vein is straight, doesn't have that hooked over, almost right angle aspect to it. We know that's a little bit tricky to tell. We'll look at it.
Which genus?
What, pardon?
Good question. Which subgenus, which genus?
Oh, this is Protandrena, and the species is Bancrofty, I believe. It's stuff, yeah, Bancrofty, which I got in South Dakota in the Badlands. And the subgenus is Protandrenidae, so it's its own subgenus.
Thank you.
Basal vein straight, not hooked, so that bumps out all those near-ish halictids. It has 2 subantennal sutures, which I believe Halictidae has only 1. I could be wrong about that, but other groups definitely just have 1. Tip of marginal cell pointed and well away from wing margin, so it's pulled away, not on the margin, which again separates it from a whole bunch of other genera. And we have a narrow facial fovea that is unlike the Andrena one, so no hairs lining it. And there is no scopa on the femur. So a lot of species have pollen-carrying hairs, scopa, on both the tibia and the femur, and even the basotarsus, and even coxa. So, and Andrena itself has that a lot. So let's look at those things here at the microscope. So a bit bigger. It tends to have, we'll get to the face eventually. This is a good place to look at the wing. I don't know if it has, it definitely does have markings on the face often. I just don't see this group very often myself to know if it has never has markings on the, it wasn't mentioned on the rest on the thorax or the abdomen. Here you can see 3 submarginal cells. So instead of the normal 2 in the other groups, does have this big stigma, but this length here along the marginal cell is more than half from here to there. And it also is not important because Perdita has 2 submarginals and Protandrena has 3. So they're separated out easily by that. We look at the basal vein here. So the basal vein, 5 minutes. Okay.
10
minutes.
Probably wrap up with 5 for last question.
Okay. The basal vein here is not hooked over strongly. There's an arch to it, kind of uniformly. If it was Halictidae, you would see it going along and then sharply bend down to whatever that vein is there. So that's a key feature that says, oh, this is not a Halictidae, it's something else, because this could look an awful lot like that group. So if we look at the, yeah,
let's move on real quick. Paula says that other subgenus of Protandrena have 2 submarginal cells, and I invited them to share some information.
Okay.
So I'll put that. I ask you to put that in the chat for now, but if we have some time at the end,
sure, yeah,
please do.
Okay. And so on the face, we have the usual subantennal suture situation. You can get this in focus. Oops. And so this little, in the species that I'm aware of, this is really similar in the females, where there's this little sort of beat patch of color right on. It does not encompass all of the Calliopsis. Now, whether that's true for all of them or not, but these are the ones that I see all have this sort of white patch there. There's the rim of the Calliopsis right here. You can see in this particular one that it's more difficult to pick out the subantennal sutures because of the topography here. Let's see if I can play around. So what we're looking for is 1 and 2, but we don't have a great, there's so many pits in that area and some hairs that we're not getting.
I think I see them better on the top one.
Oh, on the top one? Okay.
Just at like, they're shimmering.
The shimmering subantennal sutures. Yeah, let me maybe I'll run this askance a little bit, and we will see if the subantennal sutures show up. But it should also have a long inner subantennal suture in addition to having 2. Not so easy to see for whatever reason. I think there's just a lot more topography going on there. So if you're playing around with your specimen, that you're rolling it in your fingers, you should be able to track the subantennal suture. Let's try the vellum paper approach. And yeah, nada. Okay, so difficult to see, but yet present. So I think that covers. So there's, I should point out that if you use, for example, the Michener's and McGinley's books and perhaps other things, they'll talk about sutures along the mesopisternum. So the mesopisternum is this big plate below the front wing, and there's a suture line that runs across. I don't even know if the Protandrena group has that, but the Panergines do. That's a character, but one that almost doesn't get used very often. It's hard to, the wing is usually blocking some of that visual, so tends not to get used. So I think, do we want to jump into Panerginus? We have it. It shouldn't take that long. What do you think, Claire?
Yeah, I was wondering if we're gonna
Or if there's any questions.
Dan actually brought up a really good point that leads nicely into oh I know we've been this progress. I'm gonna bring this up real quick. In the B Genera of Eastern Canada, Packer et al. Wrote Packer et al. We get to Protandrena when we choose 2 submarginal cells. In fact, with my specimens, when the key brings me to Protandrena, we are rather having pseudopenergus. Any explanation to that?
So in the Canadian guide, so if I get you right, if you're going through the Canadian Lawrence's guide and you click 3 submarginals, what do you get?
I'm going to ask you, Sam, to turn on your speaker, and Anne to.
Okay. All right. My speaker is on.
Okay. So Anne, if you want to unmute.
Hi. I'm a French speaking person, so I'll try to get it in English. So in my lab, we often work with the paper from Packer and Hall, the B genera of Eastern Canada, to get to our genera. And that paper, which may be old or too old, and maybe there have been some changes since the time it has been written, but Protandrena is said as two submarginal cell species, and in fact our specimens that with decay comes to Protandrena genera are not Protandrena when I work with the other two, and they are pseudoparnergus. So just want to know if there has been changes during maybe the last decade with those genera.
So I'm not, I'll have to talk to Lawrence about that, but I am not, I'll have to look into it too. So I think theoretically, particularly in the East, you would only have probably Protandrena abdominalis, I think, up there. Maybe there's another northern one that sneaks up. I thought they all had 3. So I'll have to investigate. Maybe there's some northern species that is 2, but I also think the Discover Life guide, I want to say, has all the Protandrena species for North America, north of Mexico in it. And so if someone wants to pop over there and look, we can do that.
2-minute warning.
2-minute warning. Okay. I can stay a little bit longer because it's too interesting. Okay, so if we go to Protandrena here, so now we're in the Discover Life guide. Oh, I forgot to look and see whether it includes every Protandrena, but I think it does. Okay, and we look at the characters. See, there's no character that is some have 3, some have 2, which would be a great character, right? If some species had 2 and some had 3. So maybe, I don't want to cast aspersions on anything Lawrence has done, but maybe that's a mistake in the guide. So I'll poke around and I'll ping Lawrence after this, or if anyone's up there in Lawrence's shop right now can run over. He might be in France still, but anyway. So that's all I know. Worthy of investigation, and we are always very interested in hearing about any possible collisions, differences, problems, better ways to explain it, or just saying like, I don't understand what you're talking about here, or I was confused, because we can change any of these characters at any time. So I guess I'll stop there because I'm not sure I have any more insight into that 2 versus 3 on Protandrena issue. Does anyone else?
So Paula's going to follow up with us on the south of the border specimens in an email. So
Okay.
That we will share it. Apparently you were very clear today, so we don't have any specific questions.
Okay. Well,
They're kind of more rare things to see.
We'll cover then the Pseudoparnergus and Panerginus at the same time, because they're often confused between the two, and so that'll give us a chance to do both at the same time, and we can contrast them. How about that?
That's a great idea. In Paula did say, in the bees of the world key to subgenus, there are 2 options: 3 for SPR and 2 for the other.
Okay.
That's strict do. I think that's what.
All right, we'll have to investigate that and see what's going on.
Okay, I will copy that. Paste that for our reference later.
Oh, I should, Claire, if I could interrupt.
No.
I'll have to say that there has been some back and forth on some of when we get into the Pseudopernergus in particular, as to if there may be, people have in the past wanted to split that into more than one genus. We're treating them right now. We generally follow what John Asher has listed out, and I'll have to look and see if he's thrown out the Anthomergus group or not. But for now, just a note for people that they may see additional genera that we are calling only subgenera at this point, but we just follow one taxonomic thing. We don't try and make taxonomic decisions ourselves, because we would just get in trouble. So we follow what John Asher puts together, maybe with a lag, but usually we follow it.
Okie dokie, I'm gonna stop recording. Sam has to jump, but I'll stay on while the recording downloads. So if you have any final things to bring up.